Okay, observation. What's the next step? Form a question. Next, research. All right, so now we're kind of starting the experimentation part. And that requires us to form a hypothesis, right? Because we've done our background research, we're beginning to kind of think about what we want to study. We're coming up with our experimental design. We want to form a hypothesis. So a hypothesis is something like an educated guess, right? You could say it's not tested or proven yet. Oftentimes it's anecdotal evidence. And as we said, at the end of the experiment, so your hypothesis is what you set up at the beginning, and you say, okay, I think that onions grow better in sunlight, right? And then you set up your experiment to either accept that hypothesis or reject that hypothesis. So remember that we accept hypothesis, we never prove them. Remember I said it's my pet peeve because you guys will be doing lab reports and some of you will still say that you proved your hypothesis, but you didn't, because there's always the ability that further testing and data show otherwise. So we use the word accept. All right, we did our worksheet. A little bit of vocab here. When we talk about facts versus theories, I just want to kind of go over that, and then we'll get back to our hypothesis and experimental design. So a fact is not really something we test in science. It's a careful measurement. It's 98 degrees outside today, or the moon orbits the earth. These are all careful observations, but they're verifiable observations. They're just things that are known to be true that aren't necessarily tested all the time. They're simple. There's X amount of people in this room, right? So that's a fact. It's a careful observation. It's verifiable observation. So a theory is something very different in science, and you might have also heard things like laws, and we won't get into that, but we will talk about theories. Theories are much different in science than they are in everyday language, right? Every day we often talk and you're like, okay, I have a theory that, every Tuesday it rains or something, right? You might use that word theory very loosely in everyday English language, but the word theory is not that way in science. In science, the word theory is something that has been elevated. It's a hypothesis that has been very successful, and so it's elevated to what we call a theory. So a theory is a highly successful hypothesis that has been accepted many, many times with lots of evidence to support it. So something like the Big Bang Theory, right? It has that word theory at the end, but don't get confused and mean that it's not just a theory. There's been lots and lots of studies that have gone to support this theory, and so it's elevated to that term. It's not the same as our English everyday language as the term theory. So theories make predictions, they can be tested, but again, the big point here is that a theory is an elevated term that we use to describe a phenomenon or something in science that has had lots of support and lots of data that has supported that hypothesis. So things like the cell theory of evolution, right? These are theories, but that doesn't mean that they're just theories. They've had lots of evidence to support them, okay? So just a little difference between a theory and a fact. All right, so back to experimentation, right? So we're on the experimentation or the create the experiment part of the group. Has everybody got their sign in? All right. So we have a couple of different vocab words, and it's helpful to give examples as we go along. So if something doesn't grab you right now, just keep working through it with me when we go through some examples. We have two main types of groups in an experiment. We have what's the experimental group, which there can be multiple experimental groups, and then we have a control or a placebo group. And then we have what are called variables. These are independent variables, dependent variables, and controlled variables. So these are things that we may change in an experiment. So I'm going to show you a video, and then we're going to work through these with an example because I think it makes more sense that way.
sentence is grammatically correct.
Independent variable and dependent variable are important terms related to controlled experiments. Remember, a controlled experiment is a scientific test in which all conditions are kept constant except for the variable you are testing. The independent variable is the thing you're testing in an experiment. It's often abbreviated as IV and sometimes called the manipulated variable because you change or manipulate this variable. In an experiment, the dependent variable is the thing you're observing and measuring, the thing you're anticipating may be affected as a result of exposure to the independent variable. It's often abbreviated as DV and sometimes called the responding variable because it responds to the change that you make. Let's look at a few examples. Suppose your hypothesis is that if students study 15 minutes a night, then they will have higher test grades than those who don't study at all. What's being changed or manipulated? It's whether or not the students study for 15 minutes. And what's going to be observed or measured in this experiment? What do you think might be different as a result of this increased study time? You're expecting test grades will be affected. In this experiment, the independent variable is the study time. And the dependent variable, the thing you're measuring or going to observe, is the test grades. Here's another example. See if you can figure it out. This time, the hypothesis is that if people who have headaches take aspirin, then they will get relief faster than those who don't take aspirin for headaches. So, what's the thing that's different in this case? The thing that's different, the independent variable, is whether or not somebody is taking an aspirin. Then, what are you going to measure? You're measuring how long it takes for their headache to go away. That's the dependent variable. Here's a final example to help you understand these terms. You predict that if a brand name light bulb is left on continuously, then it will burn longer than a bargain brand light bulb used in the same manner. In this case, what is the independent variable? It's the brand name light bulb. What are you measuring? You're measuring how many hours the light bulbs work before burning out, which is the dependent variable. The independent variable is the brand name light bulb, and the dependent variable is the amount of time the light bulbs work before burning out. So, to review, the independent variable is the thing that you're testing. Sometimes this is referred to as the cause in an experiment. It is also the if part of your hypothesis. The dependent variable, the thing you're measuring, is the effect. It is also the then part of your hypothesis.
Bring your own phone. Well, your own real phone when you switch to Google Fi, a phone plan by Google.
Okay, so we have the...
Hi everyone and welcome back to High School Science 101. Today I am
So we're forming our experiment. This is for a typical controlled experiment. Not all science, not all biology is this pretty nice and perfect. I told you I worked in wildlife management. We work in the field a lot. We don't always have nice, pretty laboratory settings. But this is a nice overview for what makes a good experiment, especially in a laboratory setting. So we have our main parts of an experiment when we're setting one up. We have our groups, which are our experimental, and our control groups. I can just say control groups. And then we have our variables, our independent variable, our dependent variable, and our control variables. So I'm going to use the example of the last one that we saw when he was talking about the light bulbs. I set it up as a graph to look at if we did that experiment and we did a graph to show the results. I'm going to point out the different variables and that might, in the control groups and the experimental group, and that might help us to understand the different parts here as well. All right, so pretend that we did the light bulb experiment. So we had brand name light bulbs and we had our bargain name light bulbs, right? So probably if we were doing this, do you think we would just choose one brand light bulb and one bargain light bulb? No, what might we do instead? Yeah, or maybe like 100 of each, yeah, like 10 of each, 30 of each. In biology, the magic number for statistical values is 30. So yeah, maybe we would have 30 brand name light bulbs and 30 bargain light bulbs, and we would run them and we would see which one lasts longer. Okay, so this is a bar graph here. This is the average of all of my brand light bulbs, and this is the average of my my bargain light bulbs for how long they burned. Okay, so here is my y-axis, y to the sky, is always going to be my dependent variable. And in this example, hours is my hours burned, I guess would be more specific. Number of hours that the light bulb burned would be my dependent variable, or the responding variable. So the dependent variable is always going to be on this y-axis. A couple ways I think of it is it's dependent, so it has to stand up on the independent axis in order to carry out, or that if you're trying to figure out which is the dependent variable and which is the independent variable, the dependent variable, the number of hours burned, is dependent on if it's a brand name or a bargain name light bulb, right? If you make it into a sentence, that helps too. So then in this example, our independent variable, our manipulated variable, is our brand name light bulb. And then our bargain light bulb, that's our control group, because that's where we're using to compare to see if the brand name works better or lasts longer. All right, and then one thing he didn't mention, but we're going to talk about, our control variable. So these are things that we hold constant in an experiment. So for this example, what are some things we might want to hold constant in this experiment?
How long we're burning them?
Well, we're going to be burning them until they burn out, but yeah.
The setting.
Okay, so what about the setting?
Same, like the same room.
Okay.
Yeah.
Yeah, it might be the same room, or like temperature
Temperature.
too, I think of. I heard something else.
Keep the power input the same.
Yeah, so the power input or the wattage, you might want to be the same. Right, you don't want to do like a higher wattage versus lower wattage because you want them to be comparable to each other. So these are things, and we'll have another example, these are things that you want to set up to be, so that none of these are actually making or affecting your results. All right, so we're going to do some more examples, but those are the main parts to an experiment, and we will keep working on this throughout the semester, but it's a good idea to try to get a hold on especially the difference between the independent and dependent variable. What might be a hypothesis I would have created before I did this experiment?
Name brand names burn longer than generic.
Sure. Brand name light bulbs burn longer than bargain light bulbs. Something like that. Or there is a difference between the two. Okay, so we're still on experimentation. So this is a review for independent and dependent variables and our controlled variables. So independent variable, again, this is the factor that is deliberately being changed in the experimental group. Sometimes we might have control groups that go along with them. So in our case here, our independent variable was the brand name light bulb, and along with that was our control group, which was untreated, or that's kind of like our bargain light bulb. Bless you.
Thank you.
The dependent variable, this is the measured result, right? So the number of hours burned is the result of the type of light bulb. And this is analyzed in both experimental and control groups. Now our control variables, again, these are the things that we want to control for, factors that we want to keep constant to eliminate some type of bias. All right, when we have an experiment, we want to eliminate bias, and there's a few ways you can do this. We don't do this too much in this apply in these labs, I don't think. But there are things, this is a lot of times, if you study like psychology, you might have heard of these studies a lot too, but maybe in medical science, you might have blind studies. And say we wanted to test the effect of caffeine on a group, right? And I'm the experimenter, you guys are the group I want to test it on. You either get caffeine or you don't get caffeine, but you don't want to know that as somebody who's being tested on, because if you know that you received caffeine, then you might mentally just feel more excited. Can you let this? So you don't know, so you're given a pill, you're given like a placebo pill or a caffeine pill, because if you know if you've received the treatment or not, your body, your mind might react differently to knowing that. So that's a blind study. A double-blind study is just like what it sounds like. In that case, I don't know what you received and you don't know what you received. It's written down somewhere, right? And the results are analyzed. But that helps prevent the bias on the experimenter's part, because if I know you received a caffeine pill or not, then I might interpret your behavior because I already knew the answer, right? So that's a double-blind study. Okay, so take a couple minutes, work with a partner or a group, and I'll read this to you and we can kind of work through the concept of an experiment. So determine if caffeine improves plant growth. A scientist waters 15 tomato plants with caffeinated coffee and another 15 plants with decaffeinated coffee. The scientist measures the plant heights of each plant for 30 days. Both groups of plants are grown under the same light, humidity, and temperatures. So in your group or with your partner, identify the independent variable, the dependent variable, the controls, and the control group. And then what is the question that the scientist is trying to answer here, and come up with a hypothesis for the study. And then we'll go over it together.
So the independent variable is caffeine. Of course, yeah.
Yes.
Oh, the independent is if caffeine improves plant growth.
With
And it looks like the dependent is like whether it's caffeinated or decaffeinated.
Two that we
And then the control is like the environment that it's in.
Yeah, the controlled variable looks like, well, yeah, it's the same amount for each.
Yeah.
So like 15 tomatoes and then,
Yeah.
yeah. The light, humidity, and temperature.
dependent is how much they grow. Control is same light, humidity, and temperatures. That caffeinated and grow more chipper.
And the amount of time too, like the 30 days.
Yeah. So time, the amount, even the environment.
And I think the control group would be the decaffeinated.
Yeah.
That was me yesterday.
That the caffeine improves plant growth.
Okay, so caffeine would be, would be the manipulated variable. Part of the experimental group would be the plants with caffeine. Okay, so the amount of caffeine itself is the variable, or the fact that caffeine is given or not. What would be the dependent variable? Growth. And how are we measuring that in this example?
Height.
Okay, so it would be, if we're really, say, they have to measure it, say we're measuring it in meters, so we would have 0 to, I don't know, 100 or whatever. So height, and we will go over this more, but you always want to have a unit and you want to be specific. So height in meters would be our dependent variable. What is our control variables?
Light, humidity, temperature.
The temperature, yeah. The time frame. And then the amount of tomatoes.
Yeah. I mean, it doesn't have to always be constant, the number of tomatoes. There are often examples when you're comparing two things where you might have more tomatoes in one group than in another group, especially, like in the field. If you're trying to collect turtles or something in two different groups, maybe you only can collect so many in one group and the other. So they don't always have to be exactly the same for this, but they can be, yes.
Would you want to make sure like all 15 available tomato plants have the same amount of caffeine?
Yeah, that would be good too. Amount. So for our experimental group, the amount of caffeine, yeah, that's a good one. Right, because you don't want to give one tomato plant more caffeine than the other. All right, good. Then which is our control group?
Do you need caffeine?
Yeah, so our plants without caffeine. And what is the question that they're trying to answer?
Caffeine, does caffeine plants grow better?
Okay, if caffeinated plants grow taller. All right, and what would be a hypothesis?
If we give tomato plants caffeine, then they will grow taller.
Yeah, if we give tomato plants caffeine, then they will grow taller. Okay? We'll keep working on this kind of stuff, but try to make sure after tonight or this weekend that you can understand and do this on your own and get through the pieces and parts. Identify the parts of the experiment, because this is constant throughout the entire semester that we have to be doing this. All right, so now we're still on to the scientific method. So now we're on to conclusion, scientific method on to conclusion. So we've done our experiment. Now we have to come up and analyze our results. Or did I miss data collection? That one. Right?
Yeah.
Okay. So we collected our data. So now, after we're collecting our data, we want to analyze our results and do statistical analysis. So we're going to work through this with lab 3, 4, and 5. You guys are going to be creating an experiment, and you're going to be working through statistical analysis. So this is to introduce you to that. We will be working through it more in lab 3, 4, and 5. The first thing you want to do, and when students sometimes they don't know where to start, they don't know how to analyze or interpret the data, the first place to go is what we call the measures of central tendency. So if you don't know where to start, you don't know what to do, how to look at your data, look at the measures of central tendency. These are the mean, median, and mode. Remember those a little bit?
Yeah.
All right, what's another word for mean? The average. So you take all your data points, you add them up, and you divide it by how many data points you have, right? That's your average. Your median, does anybody remember what the median is?
It's like the middle.
Yeah, it's the middle value. So if you put all your data points up, if you put the height of all your plants in order, so say we had 1, 2, 3, 4, 5. We had 5 tomato plants. One was 1 meter tall, another was 2, another was 3, another was 4, another was 5 meters tall. Our median would be 3 meters tall. All right, our mode is the number that we see most often. So I'm going to have new data. Say we had 1, 2, 2, 4, 5. Our mode would be 2. All right, and then after you do measures of central tendency, then you can start with statistical analysis. And we will do a little bit of that in a couple of our labs. And this helps you to learn if there is a statistical difference between your two groups, okay? So right here, for my example with the plants with caffeine or not, just looking at the averages, which one is, are they different? Yes, they're different, right? So one would easily say, if you're just looking at this data, oh yeah, plants with caffeine, they definitely grow taller, right? But this is just looking at the average. There's no statistics that have been carried out in this example. So you cannot say that this applies to you can't have statistical confidence. You don't know if this is just by chance, if this just happened this one time. So in science and biology, you want to try to have statistical confidence so you can be more confident that your answer applies to the entire universe. So you might do statistical analysis to know that there is a statistical difference between the two. You can be 95% confident or more or something like that. So we will talk about this more. What you guys will be doing in your lab is you're going to be creating something called error bars, and this is kind of like statistical analysis. This helps us to see if there's a big difference between them or not, and we'll get to that. I just want to introduce you to the concept right now. But there's a difference between something looking like there's a difference and there actually being statistical differences between them. Part of what makes something statistically different, or when you have confidence, is the sample size, right? So if we did our tomato study and we did 10 plants each versus 1,000 plants each, which one do you think we could be more confident in the results? 1,000, right? Because 10 each, right? That could have just been due to chance. Maybe something else happened. So the number of experimental subjects, or the number of times an experiment is repeated, is our sample size. All right, raise your hand if you have something with caffeine in it today. 1, 2, 3, 4, 5, 6, 7. Oh, not a lot of you actually. How many of you got less than 8 hours of sleep last night? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, like 20 of you? Okay, so a lot more of you. How many have taken a college level science class before? 5, 6, 7, 9 or so. So can we conclude so if we were to graph this data, would we be able to draw a correlation between caffeine, hours of sleep, and how well you do in science class? Can we conclude, and can we call this, if we did a survey of this class, would we be able to expand it to all of ACC, all of Colorado, all of the entire world? Or it depends on the sample size. If we just sampled this class versus if we sampled the entire school, et cetera, et cetera. So statistical significance gives us a measure of confidence. All right, so we did this example here of the effect of caffeine on memory. These little blue dots, those are our data points. We have 1, 2, 3, 4, 5, 6, 7, 8. So it looks like we have 8 data points. They looked at a memory test, the score, the intake of caffeine. What does it appear to look like? What do the results appear to look like on this first graph? Yeah, it looks like the more caffeine you have, the better you do on a memory test. There was 8 data points. Can we be very statistically confident? No, probably not. Here, there's 100 or so on there, right? And it looks like there's a larger, we can be more confident in our results because we have a larger sample size. So the idea here is the difference of correlation versus causation. Remember that phrase, correlation does not equal causation? So just because something is correlated doesn't mean that there's a cause. So the takeaway message is that correlation does not imply causation. In this example, right, we have 1, 2, 3, 4, 5, 6. Looks like the higher the IQ, the more you have a lower IQ if you sleep more. You think that makes a lot of sense? Right? Maybe, maybe not, but we have like 6 data points here. So we're probably not confident in our results. Yes, somebody measured this and they saw that higher IQ and lower sleep went together, but we're based on 6 data points here. So we cannot be totally confident. There's a correlation, but there's no causation. We don't have enough data. All right, so observation, question, research, experiment, data collection, conclusion. Now we're on to sharing results. So what did we say? We talked about this a little bit, right? What does it mean to share your results with with anybody? What are some examples?
Publishing.
Okay, so publishing it into a peer-reviewed journal. How else can you publish your information?
Blogs.
Blogs, yeah. We live in 2019, right? There's blogs, the news, right? The media, social media.
Facebook.
Well, and so I told you I work outside of here. I do a lot with rattlesnake research here in Colorado, and we do a lot of social media education to the public about our results and what we know and safety and what they're doing this time of year. So a lot of our research is actually for the public, so we do use social media a lot. What else? How else? Who else do you share your results to? Yeah, your peers, your professors. So we're sharing our results, and this is really, really important. And this is something biologists are still trying to figure out how to do better. We talked about on Tuesday how sometimes it can be hard to share your results because, as we know, science is ongoing, biology is ongoing. Just because you answered a question in one experiment doesn't mean that you know everything, and so sometimes people have a hard time that if science says they don't know something exactly, then they just want to poo-poo the whole thing, right? But we know that we're going to learn how to read science and understand it so that we can be better informed, and we can either accept science sometimes or not based on what we think about the study. All right, and then lastly, again, you're never done. You're going to have a new question or carry out the experiment again. Science is ongoing. It's never ending. We might have a question and we might spend our whole life, right, trying to answer that question. So it's hard to always have one precise answer because we're evidence-based and we rely on data. It's not always easy to have one question, or you want to move on and you answered your question and now you want to ask another question related to it. All right, so scientists must share their results, right? We talked about publishing, collaborating with partners, with the media. Oh, another one that's big, it's really important, is with the government, right? Because the government makes policies, the health department, the environmental protection agency, right? All of these different departments within the government, they should be relying on scientists, climate change, right, to help them with policy about emissions or environmental impacts, right? They should be relying on scientists, and scientists need to help them, government officials, to do their job, because the government officials or the senators or representatives, they're not scientists, but they rely on scientists, or should, to help them with certain policies. Okay, and we're gonna spend some more time, I think, in lab 4, well, in our scientific activities, not lab 4, it's next Thursday, on peer-reviewed literature versus the news. So I'm gonna just kind of blow past this right now, because we have like a whole time devoted to this next Thursday. Okay, so we'll take a couple minutes, work with a partner or a group and answer these questions, and then we'll go over them together. This kind of wraps up our scientific process, and then we're gonna move into like evolution really quickly. Okay.